Hi guys, after a bit of a break, I'm back and so we'll get started, okay? The second part of this course starts with a discussion about how it's the information in DNA that is used to make mRNA, and then how the sequence of bases in mRNA is then used to insert, using the ribosome and the transfer RNAs, insert the right amino acid onto the growing polypeptide chain. And in that way, we get the right sequence and combination of amino acids in a polypeptide chain. Because of that, we get the right R groups in all the right places in the polypeptide chain to form the right types of R group interactions. Remember, disulfide, ionic, hydrogen bonding and hydrophobic interactions. And because that all happens in the right way, we get a protein that has the right tertiary structure. And because we've got the right tertiary structure as shape, that protein can then go on to perform its right function, which might be as an antibody, it might be an enzyme, it might be a membrane protein acting as a channel, it could be an ion pump. By this stage of your course, you've been exposed to lots of different types of proteins, and I guess there is an assumption that you appreciate that all the proteins, all those enzymes, all those channels, all those different types of pumps, all those ATP synthases, the carrier proteins and photosynthesis and respiration, all those things, all those proteins are there doing their job because they have the right tertiary structure, because they have the amino acids in the right place, and those amino acids are in the right place because the sequence of bases in the mRNA is right, and the sequence of bases in the mRNA is there because it's been copied from or transcribed from the sequence of nitrogenous bases in the DNA. So it's the correct sequence of bases in the genes that allows these proteins to have the right shape, and it allows those proteins to then do what they're supposed to do. So I think there's no part of biology where we can actually escape the fact that it all comes down to proteins having the right shape, and that the proteins have the right shape because the bases in the DNA are correct. And if you do need to go over that, there is a video that I've made on that called nucleic acid functions, or functions of nucleic acids. So please review that so that this next thing is going to make sense. We are now going to talk about mutations. Okay, so mutations is essentially a spelling mistake. Okay, there's been an error, there's been some kind of change in the order of bases in DNA, and that can lead to a change in the order of amino acids, and that will then affect the function, it will first affect the shape of the protein, the tertiary structure, and if it affects the tertiary structure, it could affect the shape. If there's an error in the original base sequence of DNA, it's going to make an error in the mRNA. That's going to lead to possibly a wrong amino acid being added in the chain, and that's going to change the shape of the protein. So we've already generally talked about mutations and their consequences already, but very briefly and maybe very superficially. And so we'll now talk about mutations. So first of all, let's just talk about what the causes of mutations can be. Okay, so the main causes are things like UV light, high energy light that can, when it's absorbed by chemicals, it can cause chemical changes. So what we're saying is that UV light can chemically alter the bases in DNA. And once that happens, once you change the bases, even just in one strand, when that strand is then replicated, during mitosis or even meiosis, when that strand is then used to make another copy, Because we've chemically altered the bases, when that strand is copied, it could result in an error, okay? So the chemical alteration of the bases in DNA could result then in a change in the base sequence. Okay, so say we alter one of the bases in the DNA. It used to be a T, but now there's been some kind of chemical change. Now, during replication, when an adenine nucleotide should be coming in and base pairing, because that chemical change, it might not be adenine that base pairs. It might be something else that might come in base pairing incorrectly because we've changed the structure of the nitrogenous base that would have normally formed those hydrogen bonds. Because we've changed that, it's not the A nucleotide that comes in now, it could be something else. And now, in the new strand that's made, because of that error, it won't be an A there, it might be something else. And now we've got, and when that strand is then replicated in the future, that new base will then be copied, and we've then added an error into our genetic code. Okay, so that's one way that could happen. UV light, other things are ionizing radiation, again, high energy radiation that, again, same thing, could cause a chemical change in one or more of the bases. And if that happens, we've already discussed, that could change the copied strand that then passes on when that strand is itself copied. Okay, and finally we have chemicals called mutagens. When these chemicals are in the cell, they can make their way into the nucleus near the DNA, and these mutagens, either they can chemically react with the DNA, again, chemically altering the bases, so either they can chemically react, or the other thing that can also happen is some of these mutagens have a structure that's very similar to the nitrogenous bases. And when replication happens, or when meiosis happens, they are mistaken for a base, which causes an error in the replication process. Yeah? So they can chemically react, or sometimes they can resemble nitrogenous bases and thus cause an error during replication if they're there or thereabouts. Okay? So these are the kind of ways that bases in DNA can be changed. I don't know how important it is for you to know exactly how this happens, but you know, if you have an appreciation of it, it's not going to hurt. Okay, so the first thing that we've covered is causes. We're next going to look at the types of changes that could happen, and their possible consequences will be following that. So now let's talk about the different types of mutations. So our first type is substitution. Now in a substitution, what happens is one of our bases, the error that happens is the base changes to another base in the same position. So in a substitution, what would happen is our sequence here on the template strand would read T, T, and instead of G, for example, it might change to T. Okay, and this would carry on and be read in the same way, no other changes. So the only change here is that G has changed to a T. Now, what would happen is that would cause the amino acids in the chain to potentially be different. So TTT actually results in the addition of a lysine to the peptide chain. CCT in this case would still give us glycine. Okay, so substitution has the potential to change the sequence of amino acids and therefore affect the function of a protein. The next thing that could happen is deletion, or the other type of mutation could be a deletion. So in that case, what would happen is our G would be deleted, and so everything would move down one. So because the G is no longer there, we have to carry on then with C, that one, T, and then whatever else might be there. So it might be a G. Okay, could be something else. Okay, so this has the potential for quite significant changes, because what happens is, whereas here we only really affected one amino acid, the deletion of one base causes so you know, TTC would give us a different amino acid, CTG would also possibly give us a different amino acid, and effectively what we've done here is we only changed one codon, because we only substituted the base, so we just changed that codon from TTG to TTT. But here, we've effectively changed that codon and every single following subsequent codon from this point. So that can result in a rather large change. So let's just confirm that in this case, what we've done. Okay, so by causing a deletion of that G base, we don't just carry on reading in the same frame, we cause a frame shift. So deletions have the potential to cause a frame shift, meaning that because we're now reading that C as part of this codon, and then that codon changes to this base, that base, and then the next base, and so on. So we've changed that TTG to TTC, CCT to CTG, we've changed from asparagine to lysine there, glycine to aspartate, and potentially every single other codon will be different and potentially different amino acids. So there's a quite large effect of that deletion right there. Okay. Insertion, similar to deletion, insertion can also have a quite large effect, because what we're saying is we could have a T right there, we could have the T, and between the T and G we could have another base inserted. So we could have here, we could have an A, for example. Actually, that's not a good idea. Let's put a C there. So T, T, C, and our G is still there. So G is there, and then we carry on with that C there, and then that C moves there, and then that T moves along. I'll just put that there. Okay, so again, we've got the potential for very big changes because we've inserted one base. Now that codon is different. That codon is now different from the original, and so will the next one be, and so will the next one be. So just the insertion of a single base can cause, again, it can cause a frame shift, because we are changing the reading frame from the point at which the insertion happened. So every single codon subsequently is affected. Okay? Duplication is simply that, the duplication of one or more bases. So we could have, right here. Okay. And duplication could be that some bases are read or some bases are repeated. So instead of T and T, we could have a duplication of that TT sequence. So we've got TT again, T, we could have T again, and then from that point on, we continue our reading frame, so G, C. So again, we have the potential for a frame shift here, okay? So if a sequence is repeated, it can cause a change in the reading frame in every codon from this point on, because this codon has changed, that codon has changed, every single other codon has changed, okay? An inversion is when part of your sequence just gets flipped over. So what we could have here is we can have our T, that's the first one, but then this portion, T, G, C, C, gets inverted. So we could have that C there, then that C, then that G, then that T, and the other T could be in the right place. Yeah? So essentially what we've done is this portion is just been flipped over and put in again. These are the kind of errors that can happen during replication. Yeah, because remember we're dealing with a large molecule. It's a very complex process. It is a kind of miracle in a way that these things don't happen more often. Yeah. Okay, so because of that, again, so now we're reading TCC, GTT, where we had TTG, this is our correct sequence right here, TTG, CCT, we've now got TCC, GTT, because of that inversion. Okay, so the amino acids that would be in that. Okay, so in that place, we would get arginine and glutamine. Okay. Right, you will have noticed that I've just kind of put an asterisk in those places simply to indicate that there is a potential for this deletion to cause a frame shift situation, which means that we don't just have a localized change that doesn't affect other amino acids or codons further on. But the potential of these three mutations right here is to cause changes from this point onwards to every single codon or triplet subsequently, okay, or the codons that are read. Okay, so these are the types of mutation that can happen. We've discussed using an example template strand code of what these changes, how these changes can affect that code, but then also potentially how they could change the amino acid sequence. So now let's talk about the severity of these changes. That has come up a few times, so you need to appreciate the kind of dangerousness of these different types of mutations and how they could be a little bit different from what we discussed. Okay, so let's begin. So, for example, this substitution, it affects a single base, and if we just begin our discussion about that, we've changed that from an A to a T, and from a G to a T, and that has caused a change in one amino acid. Now, that one amino acid might be a really important one in the active site of an enzyme, for example, or it might not be very important in the grand scheme of the whole structure of the enzyme or any other kind of protein. So, you need to be aware that this one amino acid change could be very important for the protein, for the tertiary structure, or it might not be very important. The other thing is, it might change from one charged amino acid to another amino acid that has a similar charge. And in that case, even if the amino acid is changed, it might not cause as dramatic an effect as if it were to change from a charged amino acid to a hydrophobic amino acid. Now that kind of change might cause quite a lot of impact on the tertiary structure and the interactions. So we need to appreciate that the amino acid change that could happen could be serious, have a lot of impact, or not have a lot of impact. On one level, the kind of amino acid that it changes into, and on another level, where in the protein that it is. If it's nowhere near the active site of an enzyme, if it's nowhere near the part of the channel protein that's responsible to interact with the molecule that's going through, it might not have such a dramatic effect. The other thing is, what is the other thing? Yes, the other thing is that the base change that occurs might not cause any change in the amino acid. Okay, so we might have, let me check out the code. We started off with a TTG. Now, if we had changed it to TTA, we had TEG, okay, so if we had changed this to TTA, we would have got the same amino acid because of the degenerate nature of the genetic code. Okay, so that's another level. Sometimes the substitution that you make or that occurs in the DNA sequence, base sequence, sometimes it can lead to a different amino acid, but not in every case, because of the degenerate. That's a term that comes up quite a lot. Because of the degenerate nature of the genetic code, more than one codon for some amino acids. Okay, so there's more than one codon for, I think, most of the amino acids actually. There's more than one codon for the amino acids, and in that case, you might make a base change that actually codes for the same amino acid, and therefore you'll end up with the same amino acid sequence, the same primary structure, and therefore you won't cause any change in the protein structure. Now another thing comes to me, which is, what was this? Forget now. The other thing that occurs to me is that sometimes your substitution doesn't just affect one codon, it can have quite a significant effect because your base change could introduce a stop codon. So I'm just going to not mess around with this too much, but just imagine that we did make a substitution here. Yes, it doesn't affect the coding, the codons or the triplets or anything like that, or sorry, the codons. But say that instead of that amino acid, we introduced a stop codon, even though it's not really changing a reading frame, but it is terminating the codings. It is terminating the polypeptide chain early, very early. It might be not early, it might be very early, it could be so late that it doesn't really make a difference. But introducing a stop codon could have a dramatic effect because it means that there's a whole polypeptide sequence that's not being added onto the protein after this point. So yes, it probably does have an impact where that mutation comes in the coding sequence, but this is quite more impactful than just changing an amino acid for something else. Okay, so introducing a stop codon can have dramatic effects. So that's just substitution. So you can see even a single base substitution can have a whole range of effects, from not changing the amino acid sequence at all to leading to the early termination of a polypeptide chain. Okay? Yeah. In which case there will be a large chunk of the protein that isn't there, and that's probably going to have a very big impact on its shape and function. Okay, so that's substitution. Now, yeah, deletions, insertions, and duplications, let's group them all together, because what we've seen is that they can result in a frame shift, which causes the change in the reading frame from the point of the mutation, which could potentially change every single amino acid from that point. So that is huge. Okay? That can cause big changes in the protein structure. There is one caveat to that. There's one situation where that doesn't happen. And that is whenever the deletion, insertion, or duplication is a multiple of 3, the effect is not so dramatic. Okay? So say we deleted one base here, and that caused a change in the reading frame. If we deleted 2, it will change the reading frame. But if we deleted these 3, the only change it would have is we would be missing the amino acid that triplet coded for. Which means that we're just missing one amino acid, everything else is read in the same reading frame because we made a change of 3 bases. And if we had deleted 6 bases, we would be missing 2 amino acids. Yes, more impact, but 2 amino acids missing in hundreds. So again, and it depends on how important that amino acid was to the overall structure of the protein. But bear in mind that deletions, insertions, or duplications of multiples of 3 might then result in less of a change in the amino acid sequence, because essentially we're just causing a change in 1 amino acid, 2 amino acids, 3, and so on. We're not causing a frame shift. We're not changing the reading frame of every single amino acid afterwards. Okay? Yeah, and inversions, again, depending on the size of the inversion, if it's something small like this, it will change 2 amino acids potentially, but the larger it is, the more amino acids will be changed, and we can make an assessment of the impact based on that. But I think we can draw that discussion to a close. We've now discussed the causes of mutations and the types of mutations that can happen. I think now we'll just have a quick look at what mutations mean for proteins and functions, and that will be that about mutations. Alright guys, so this is where we are now. We've spoken about mutations, what can cause them, we've talked about the different types of mutations there are, and we've also had a look at how severe the consequences or the effects of those mutations are. Okay? We're now going to look at mutations in a kind of wider context. Okay? So, in terms of the whole organism, in terms of evolution, what are the consequences of these mutations? So we've looked at essentially how they can come about, what effect they can have on protein structure, but now we're going to look at what that means for essentially organisms, populations, and the change of those organisms over time. Okay, so what is the context? The context is, and I think this is for your understanding, but this next bit is all about how you can appreciate the role of mutations in all the other parts of the biology that you're going to do. Okay, so one thing with mutations, one aspect is going to be the mutations that then lead to a protein not working properly. So that's obvious. We've already talked about how that's going to happen. So if a mutation causes a protein not to work properly, it can, and that is going to be a negative mutation. Okay, so a mutation that is negative in its effect, and that essentially is what causes genetic disorders. Okay, so things like cystic fibrosis, things like sickle cell anemia, that's what we're talking about here. So genetic disorders. So, and so in cystic fibrosis, we have the CFTR ion channel whose function is affected negatively by a mutation, and that causes ions not to move across epithelial cells properly, and that causes the normal mucus to become much more viscous. And when that mucus is more viscous, it affects a number of different things in the body. The respiratory system, the digestive system, the reproductive system. Okay, and that's all because one protein is not working properly because there's been a mutation in the genetic code. Okay, so cystic fibrosis. Another example is sickle cell anemia, where there's a mutation in the gene for hemoglobin, which causes hemoglobin molecules to interact with each other in a very strange way, because one amino acid is kind of exposed to an environment that it shouldn't be, which causes hemoglobin molecules to effectively stick to each other, and the red blood cell changes shape because of this, and it is less effective in carrying oxygen around the body. Okay, so so we have two examples of where mutation caused a negative effect on the protein function. Okay, via a change in the tertiary structure. Okay. But the other thing is that sometimes mutations can have not so negative effects. So here we need to talk about neutral/positive mutations. Yeah. Any changes can also be positive. So, for example, if we have hemoglobin which has a certain oxygen binding capacity, okay, now that is simply because there's been gradual changes in the gene for hemoglobin over time that eventually give us a protein which is very good at binding oxygen. Yeah? So it's got all its soluble amino acids on the outside, but I think we have to imagine a situation where hemoglobin at one point wasn't very soluble. It might have been less soluble because, by random chance, it ended up with, or originally it might have had some hydrophobic or non-polar amino acids on the outside. And therefore it couldn't do its job very well because it wasn't that soluble. But we got mutations. Some of those amino acids got changed because of errors in the DNA, mistakes in the DNA. Amino acids got changed. Non-polar amino acids on the surface got changed into polar amino acids or soluble amino acids, and therefore hemoglobin became much more soluble protein as a result, and therefore was able to do its job much better of transporting oxygen in the body. Okay, so what we have to appreciate here is that positive mutations, or mutations that have some kind of positive effect, give a selective advantage to the individuals that carry that mutation. Negative ones put these individuals at a selective disadvantage. Okay, so that means that whenever a mutation is negative, it means that the person who bears that mutation has a reduced chance of survival compared to the other members of the population. And this is really important. Okay, so yeah, I guess we could put here reduced chance of survival, and remember, passing on genes. Okay, right. We could argue that this is less of a thing now, but you know, when we didn't have healthcare systems and procedures in place to deal with genetic disorders, at that time, genetic disorder would have been causing that. Okay, but positive mutations give a selective advantage, and these individuals have a greater chance of surviving and passing on genes. Right? So I think we're kind of going back to some of the things that we might have studied when we were doing evolution, and we kind of just mentioned, oh yeah, there's a mutation, and mutation gives a selective advantage, right? And then we didn't talk more about it, but I think now we can see that in context. Yeah. So yeah, mutation can give a survival advantage, a selective advantage. These individuals have a greater chance of surviving and passing on their genes. And in that case, in the next generation, there'll be more individuals with those copies of genes, those alleles. Okay? So I think the other thing is mutations lead to the creation of new alleles, new versions of genes. Okay? Did I spell that correctly? I'm not sure. Doesn't matter. Okay, so new alleles made. Now, if those alleles code for if that new version of the gene is, not as good as the previous one, we're going towards this direction. But if it's a positive one, we're going towards that direction. Meaning we've got a greater chance of survival passing on genes, and I think then, and both of these are involved in evolution. Changes which are not so good don't pass on their genes. They're not selected, and off they go. But then, if we are moving in this direction, that means that those genes are, provide an advantage. And in the next generation, we have more of those genes. In the next generation, we have even more. And over many, many years, then we get, gradual change. Okay, but then in the middle, we have these neutral ones. Yeah, so neutral ones can be either. Neutral mutations can be negative or positive, and it essentially just depends on what the selection pressures are. So, for example, we could have bears. Okay, now say if all the bears are brown, okay, in the forest, but some bears, or one bear, or, just a few, mutate. They mutate, and now they don't have brown fur, they have white fur. Okay, now this could be seen as a neutral mutation because it might not have any effect on the survival chances of the bear. But if that mutation means that the white bear in the forest is less camouflaged, surrounded by the brown of the trees, the brown of the fallen leaves, etc., no longer camouflaged because it's white, that is then a negative mutation and is going to cause it to have less of a survival chance. But say it becomes very cold, there's a change in the climate, or the bears move to another area where it's snowy, it's white. Now the white bears have a camouflage. Now they have a selective advantage over the brown bears, and suddenly that mutation is no longer neutral or negative, but it's positive. I don't know if that confuses the issue, but I think it's important to see that there is a scale of these things, and it does depend on the conditions, the environment, yeah, whatever the selection pressures are. Okay, confusing the issue a bit further, but it depends how you see it. On one hand, you might see it as confusing the issue. On the other hand, it might just say to you, or it might just show you how amazing biology is. Sometimes what we consider to be negative, and it kind of links into these neutral mutations, sometimes what we consider to be negative is or can be a positive, and it just depends on what the conditions are. For example, the mutation that leads to sickle cell anemia, we've talked about it briefly, and overall it looks like the hemoglobin is kind of less well adapted at doing its job, i.e. it's not so good at transporting oxygen. But this same mutation is what gives resistance to malaria, because the hemoglobin, the red blood cells that have hemoglobin affected by this mutation, yes, they are not so good at transporting oxygen, but at the same time, it gives them resistance to malaria because the parasite cannot reproduce in the red blood cells of the affected individual. So this gene in certain situations gives resistance to this disease, and therefore it can be considered a positive as well. Okay, so yeah, I guess either it confuses you or it just goes to show that mutations, you know, they're not black and white. They can be in the middle depending on the circumstances. So I think then this is everything, yeah? So mutations, what causes them in the first place, the types of mutations, how the mutation can cause effects on the protein structure and how significant or extreme they are. And then finally, if there are mutations, sometimes they can be negative, in most cases they will be, sometimes they can be positive, and that's what leads to, you know, evolutionary change ultimately. Sometimes they can be neutral, and their effects can only be seen if the environment or conditions change. So that is mutations, guys. Any questions? You know, I'm kind of making this up as I go along, so if I've missed anything, do let me know. But yeah, that's that. Okay, good luck. Any questions? Leave me a message.